In past chapters, we've talked about DNA and genes and how genes make proteins and how proteins determine how an organism appears and how it behaves. Now we're going to talk about how those genes are inherited. This is Chapter 12, Patterns of Inheritance, or Genetics. In order to understand genetics, you have to understand the vocabulary. We'll start with trait. A trait is an inherited characteristic that can be observed. So that would be eyes, hair, height. Every trait has two genes. One gene is given by the father, the other is given by the mother. Genes usually have more than one version. An allele is a different version of a gene for the same trait. So our example in the picture, the allele is brown for brown eyes or blue for blue eyes. A dominant allele is the allele that controls the trait. If the organism has a dominant allele, it will always be observed. In our picture, the color brown is dominant. Dominant alleles are written as capitals. The recessive allele is the allele that is only seen if paired with itself. So in our picture, blue eyes are recessive. You have to have two recessive genes in order to see the blue eyes. Recessive genes are written as lowercase letters. A homozygous gene is both alleles are the same. So we have two capital B's or two small b's in our picture. Each parent is going to contribute one of these alleles, and again will be written as two capitals or two lowercase. Heterozygous, both alleles in the organism are different. Again, each parent contributed one of the alleles, and it will be written as one capital and one lowercase letter. In this case, heterozygous means that you have a dominant allele, and so you will see that dominant trait. The genotype is the genetic makeup of the organism. That's all the alleles that make the organism look and behave the way it does. Phenotype is the way the organism looks or behaves. So genotype is the genetic makeup on the inside. Phenotype is what you see on the outside. We're going to start our study of genetics with the work of Gregor Mendel. Mendel was a monk who worked in the monastery's gardens. He experimented with sweet pea plants to determine how the trait of color was passed from one plant to another. Mendel began his work studying the color of the flowers. He had purple and white flowers to work with. Those flowers that passed their colors on to their offspring were called true breeding plants. The flowers that he began with were called the parent generation. So his parent generation had one true breeding purple plant and one true breeding white plant. The first generation of offspring, or the F1 generation, all had purple flowers. The white color had, for this generation, disappeared. In this experiment, Mendel moved the pollen from one flower to another himself, so he knew that no mistake had been made. In the next generation, he allowed the plants to self-fertilize, meaning he didn't do anything, he let them drop the pollen from one flower into another. No matter how many times he conducted this experiment, the F2 generation always had a 3-to-1 ratio. There were always 3 purple to 1 white flower. The white flower that had disappeared in the F1 generation had reappeared in the F2 generation. Mendel explained his results with his idea of the principle of segregation. He said, 2 alleles for a gene separate during the formation of gametes and are rejoined randomly during fertilization. To understand this, you need to go back to meiosis. What he's saying is, for each chromosome, you have two versions, the mom version and the dad version. They're joined up randomly, remember, during synapsis, and they're separated randomly during meiosis 1 and then during meiosis 2. Depending on what sperm matches to what egg, they are rejoined. Again, that is random. That randomness then determined whether he got a purple or a white flower. What Mendel observed was that 3 of the 4 plants were purple, which was the dominant form, and 1 out of 4 of the plants were white, which was the recessive form. So he's only comparing dominant to recessive, which is why he got a 3-to-1 ratio. But what he actually saw was 1 homozygous dominant plant, 1 homozygous recessive plant, that's the white one, and 2 heterozygous plants. That's how he got the 3-to-1, because remember, you only need 1 dominant allele for that trait, which in this case was purple, to be seen. A way to predict the possible genotype and phenotype of an organism's offspring is to use a Punnett square. When we're looking at a single trait, we're going to do a monohybrid cross. A monohybrid cross uses a box that has 4 squares in it. Mom's information is traditionally put on the left, and Dad's information is traditionally put on the top of that box. To fill the box in, we're going to start with Mom's information. We're going to use that red X, and we're going to put it in the top 2 boxes. We're going to take Dad's X and put it in the same boxes. So if you look at the top left box, you'll have XX, that's the genotype, and phenotype is female. The top right box, the genotype is XY, and the phenotype is male. You can use a dihybrid cross to predict how 2 traits are inherited. In this case, you'll have 16 squares to fill. If each parent is heterozygous for both traits, there will be 4 possible phenotypes. The pattern is always the same, 9-3 to 3-to-1. This is an example of Mendel's work with 2 heterozygous parents. As you can see, we have Mom's information on the left, Dad's information on the top, and we have 16 boxes of possible offspring. In Mendel's work, he studied shape and color of the sweet pea seeds. Dominant shape is round, recessive shape is wrinkled. Dominant color is yellow, and recessive color is green. Both parents were heterozygous for these traits, and as you can see, both by the genotype and the phenotype, we ended up with a 9-3 to 3-to-1 pattern. In order to fill in a dihybrid cross, you'll enter the parental information using the FOIL process, and we will do this together in class. Mendel explained the results of his dihybrid cross experiment using his principle of independent assortment. This principle has 2 conditions. First, the allele of each gene must be passed independently. This means the offspring has the equal likelihood to get the gene from Mom or the gene from Dad. In this case, it was equally likely that the offspring got the round seed allele or the wrinkle seed allele. The second requirement is that the allele pairs are also separated independently, meaning that it's just as likely for the offspring to have a round and yellow seed as it is for it to have a wrinkled and green seed. If you want to know if a particular genotype is possible, you'll use a Punnett square. If you want to know the likelihood of that genotype occurring, you're going to need to use probability. You'll either use the rule of addition or the rule of multiplication. Most probability problems that we will go over in class will be solved using the rule of multiplication. The rule of addition is generally used in support of those problems. We will do a number of these probability problems in class. In some types of inheritance, there are no dominant or recessive alleles. Instead, the alleles combine to create a new trait. One of these is incomplete dominance. If you look at the picture, we're going to begin with a red flower and a white flower. In incomplete dominance, because we don't have that dominant or recessive allele, we're going to start by choosing a letter to represent the trait. In this case, we're going to choose C for color. The alleles for color, red and white, are written as the small letters at the top. Our parent generation has one red flower and one white flower. Together, they create a pink flower. Red plus white makes pink, so all of the flowers in the first generation are pink. If you take two of these pink flowers in an F2 generation and cross them, you get a red flower, a white flower, and two more pink flowers, a 1 to 2 to 1 ratio. Another type of inheritance that doesn't involve dominant or recessive alleles is polygenic inheritance. A polygenic trait has a large number of alleles, each contributing a small but equal portion of information to the final trait. This creates a wide range of observable possibilities for that trait. If you think about hair color, specifically brunettes, when someone says they have brown hair, you have a wide variety, from really dark blonde to almost black. This is the result of polygenic inheritance. Not all traits have two alleles, just a dominant or a recessive. Some have 3 or more, and one of those is blood type. Humans have 3 alleles for blood type, A, B, or O. A and B are both dominant, O is recessive. So a person with an A blood type can be AA or they can be AO. A and B are also called codominant, meaning that they are equally seen when seen together. So you can also have an AB blood type. Even though there are 3 options for blood type alleles, you can still only have 2 of them as a living organism. Most alleles have only one effect on the phenotype, meaning they give you brown eyes or brown hair. There are some alleles that have more than one effect on the phenotype. These are called pleiotropic alleles. The effects of pleiotropy are difficult to predict because a gene that affects one trait often performs other unknown functions. My example is sickle cell anemia. Blood is supposed to carry oxygen in that round indented part in the picture. Sickle cells are odd-shaped, meaning the oxygen doesn't hold properly. They're also very fragile and break apart easily. Because of this, the blood doesn't carry oxygen efficiently. In addition to this, people with sickle cell anemia are often tired, they have pain in their joints, and their hands and feet swell. But an additional effect are those people who have sickle cell anemia are also immune to malaria. So we have one phenotype deficiency, which is this lack of ability to carry oxygen, while at the same time we have a phenotype benefit, which is that immunity to malaria. Your genome is your DNA, those alleles that we've talked about that tell you what you're going to look like and how you're going to behave. But those genes can be modified, and that modification comes from the epigenes. Your epigenome are the chemicals that mark your genome. It tells it what to do, where to do it, and when to do it. These are the things that make you unique. The epigenome determines the type and the amounts of proteins that are made. These modifications, in turn, determine what kinds of cells are made, how they look, and how they act. We talked about them some when we talked about DNA methylation and histone modification. Epigenes can be passed to the offspring through egg or sperm as chemical tags on the DNA and on the histone proteins. If that epigene turns one of the parental genes off, the offspring will experience what is called genetic imprinting. Genetic imprinting means for that particular gene, the offspring is haploid. It operates as if it has only one gene for that particular trait. And we will talk about genetic imprinting in the next chapter. Just as epigenes modify genes, epigenes themselves can also be modified. For humans, your epigenome may change based on what you eat or drink, whether you smoke, what medicines you take, what pollutants you encounter, and even how quickly your body ages. The study of epigenes is really new in relation to a lot of the other studies of genetics, so we don't know a lot. Currently, most biologists believe that any changes in your epigenes are probably harmless. But some early studies are indicating that changes in the epigenes may lead to or change cancers, may lead to diabetes, may lead to other types of diseases, or may increase the severity of certain diseases that are already present. This is the end of Chapter 12, Genetics.